quality and control. Functionality, I will touch on this a little later, as they consist of a bilipid layer, tiny little bubbles, and this bilipid layer, for instance, get easily destroyed if you freeze dry or lyophilize your exosomes, and therefore rendering them non-functional. You want to be sure that storage stability is given. Very important as for testing and documentation. It is critical to have third-party testing involved with your products, because otherwise you just take the word of the vendor, this to be all right, but you don't have an unbiased, approved and certified quality control that this is all okay. Regulatory compliance is also a very important part. Interestingly, for as long as we're talking about aesthetics and cosmetics, there is not so much to be observed strictly, but there are certain guidelines and laws, and our company is rooted in regenerative medicine. So all our products are manufactured according to FDA standards that govern the production and manufacturing of medical grade products. We find that important because even if just for aesthetics or cosmetics, you put it on your face, it gets absorbed by your skin, so it will somewhere enter your body. Not to mention if you have a microneedling done first, and then basically your face is an open wound and you apply those exosomes. You really want to make sure that they have ideally medical grade and they are clinically okay to be used on your skin or on your body. And you want to watch out for scientific support. Never ever buy from a vendor who doesn't do their own science and research. Also very important, never buy from a vendor who doesn't manufacture their own stem cells to actually have these exosomes derived from them, because vendors that purchase their stem cells somewhere else, again, they give up control. They don't know where exactly they come from and what the quality and potency of those stem cells are or might be. I don't want to go into this too much. I'm happy to send the presentation or the slides to anyone who is interested, but just to give you an idea, exosomes stand for precision. So exosomes are a fraction of what's called extracellular vesicles that are secreted by virtually all cells. And the exosomes are the ones of a particular size that we are interested in. They are responsible for cell-to-cell communication and regeneration potential. They exert the stem cells' benefit when it comes to regeneration and repair. Other vesicles, like microvesicles and so on, they have different tasks and responsibilities. Some of them are actually just used to take out junk and trash from the cell to clean out, and you don't want to have those mixed up in your products. So make sure that it's really purified exosomes. Therefore, purity and consistency is very important. You want to have some proof of efficacy. Conditioned media and non-specific EVs cannot do that. There's like nothing you can scientifically derive from whatever happens using them as a aspect of proven efficacy, because results really matter. Sorry. And this is what both physicians and patients look for, right? So you want to have some track record of proven results and efficacy. Inconsistent efficacy is given when you don't isolate and purify and really define your products. Questionable safety is, of course, one of the most critical aspects of it all. So if there is not a coherent proof and a certificate of conformance, you don't want to touch those products. Be aware of misleading marketing. Basically, every vendor will tell you that they have the best, and it requires some education and some common sense to really look through their marketing slogans and look a little bit behind. And I hope that I can give you some information that will help you to actually ask good question, and to discern a little bit better of what might be a good exosome and not so much. Potency is a big deal. Some don't care about any purification or characterization. They just take conditioned media or whatever products. They mix in a kitchen sink of amino acids and vitamins and peptides in order to make it more potent. All of that is highly questionable and really necessary. You, if you have a good and excellent, you don't get it. Um, if
لا ماما اللي بتطبخ.
there's somebody
بس انا ما
not muted, if you
اصل خايفه انا
لا
اقرب
انا بالعاده بفوت
تقفل عليك برضه.
هو بيقفل علي
um,
كمان.
السلام عليكم.
so also make sure that you have some scientific validation of what you're buying. If your vendor cannot provide anything in this regard, you better stay clear of it. About exosomes. So here's a nice depiction of the hallmarks of exosomes. So they have many different tasks, and their tasks are actually derived from the cellular origin they are derived from. So I like to always give this extreme example. As I said, virtually all cells secrete exosomes. You want to make sure that you use the right source. An extreme example would be cancer cells, tumor cells. They secrete exosomes too. But if you were to take those exosomes, they have a wholly different task. They are supposed to support tumor growth, etc. So that's something obviously you don't want to be touching. Therefore, as I said, stem cell are the natural best source for exosomes when it comes to regeneration, repair, and even rejuvenation. And to make sure that you really tap only into those functions that you are looking for. And even when it comes to stem cells, there are certain things that you want to stay clear of. There are the so-called pluripotent stem cells. There are embryonic stem cells and iPS cells that arguably are the most potent stem cells. They can give rise to any given cell type, and that sounds great, but they've also been found to actually be hard to control, and they cause benign tumors called teratomas when transplanted or infused into animal models. So they are not suitable for any kind of medical application, and therefore we also wouldn't want to use them to derive our exosomes. What we do want to use is multipotent stem cells or tissue stem cells, in particular what's called mesenchymal stromal stem cells that you can basically harvest from a lot of different tissues in the body, bone marrow, adipose tissue, and perinatal tissues, such as placenta, umbilical cord or umbilical cord blood, and amniotic fluid. Obviously, perinatal tissues are the prime source for stem cells and for our exosomes, because these stem cells have just finished building a baby. So they are primed to actually generate tissues, regenerate and repair in case something goes wrong in the course of development. They can fix these problems. So these are the stem cells that we want to use to derive our exosomes, because they are having the best potency, and they are also considered the safest because, as I said, they just finished building a new baby that's healthy. And even among those perinatal tissues, there are differences. And the one that has been found to be the best source for our is the so-called Wharton's jelly. That's the gelatinous substance that gives the umbilical cord its crunchy firmness. why Wharton's jelly, they also contributed to just building a new human. And what's also very important and interesting is that the Wharton's jelly is protected from maternal contaminations. So you won't find any maternal DNA in there. Or sometimes we get questions about vaccine residues, either in general or in particular with regard to COVID vaccines, that people are a little bit fearful of what these vaccines might do or the vaccine residues. And these residues, because you can imagine that basically every mother has been vaccinated in their life. Even COVID has like a 80, 90% penetrance these days. So you can assume that all mothers have been vaccinated, but any vaccine residues that she might have in her body will not make it in the Wharton's jelly. Therefore, it's not only the most prolific and most potent tissue source, but it's also the safest one. Better than amniotic fluid and placenta, for example. Bone marrow and adipose tissue, I would advise to dismiss as a source. I know that a lot of vendors resort to, donors in their 20s, and they either donate bone marrow or adipose tissue. And when we think of a 20-year-old, we consider them to be in the prime of their life, provided they're healthy and so on. So it sounds like a great source, but it really isn't, because even those people have been subjected to 20 plus years of environmental hazards like chemicals, toxins, bad food, disease, radiation, all these kinds of things, and they impact the quality of what stem cells they actually have left, which are actually not that many when you're in your 20s. You might even have accrued mutations in the course of your short life. So it's not an ideal cell source or a source for your exosomes. This is another reason why perinatal tissue or Wharton's jelly, MSCs, and exosomes derived from them are preferable. It's just to give you an overview. You don't have to get into the detail, but if you look into interesting factors, there are plenty of those in Wharton's jelly, MSCs, and therefore the exosomes derived from them, while other tissue sources, adipose tissue, bone marrow, or dental pulp, have only a fraction of those factors that are important. And many of those factors play actually a role in the exosome's
Hello,
function
how are you?
if you want to, if you're aiming at regeneration and repair. Now let's look into a few, competitors' products. So this is an example. So I won't name any competitors' names, but you will come across these things if you have a look. Check for the product ingredients. So this is a very nice example. You have here an example of a list of things that are contained in your product. And I give them kudos for being so precise to list them all, but it's also highly questionable why they are in. So, for instance, there are ceramides added that you actually don't need if you have exosomes because they contain loads of ceramides. Then, if you pay close attention, the first line it just said conditioned media. So anything is in there, and it's highly questionable if you need or want these kinds of things. There are tons of chemicals in there that are used to either stabilize or, fill these products. And on the other hand, when we talk about our H0 exosomes, this is simply just H0 exosomes and saline solution and nothing else. And this is the gold standard. This is what you want to see. If you buy exosomes, then it should be only exosomes and nothing else. All exosomes are not created equally, and there's a few things that you have to watch out for. I briefly touched on this before when we talk about lyophilized exosomes. You find them in the market a lot. Actually, the two market leaders sell you lyophilized exosomes. There is a problem with them. I mentioned it before. Exosomes are tiny little bubbles that have a bilipid outer layer. And when you freeze dry your exosomes, that layer gets destroyed. And I will touch on it again why that is important a few slides later. But for now, just be aware that there are no exosomes left. So we did a study, and I prove it to you a little bit later. We did a study that demonstrates that there's absolutely no exosomes left. So if you destroy the exosomes, all you have left is exosomal debris and trash. And among them, there might still be a few growth factors that have a palliative, which means superficial and temporary, very short-lived benefit, but they cannot function as exosomes, as I will explain a little later. Even more concerning is I did some math. They used to sell 5 billion lyophilized exosomes, these market leaders. They can no longer say that because if you say you sell that there are actually exosomes in there, and they can't. So ever since they just say 20 milligrams of lyophilized exosomes and not a number. I did the math, and when you have 5 billion exosomes, they come down to a wet weight of about 3 microgram. And if you compare the 3 microgram with the 20 milligram, it means that only 0.0152% of what's in that vial is exosomal debris or exosomal material. The rest is additives and fillers, cryoprotectants, and other chemicals, which make out 99.98%. So when you buy these, you basically buy almost 100% exosomal trash with no functional exosome left inside, which is a clear sign that you might want to stay away from those. There's another thing out there that's called hybrid exosomes. The term, which is used by one vendor, which is also one of the market leaders, they used to sell bone marrow-derived exosomes, and then they figured out that umbilical cord-derived exosomes are actually much better as a source. And they came up with this genius marketing idea to just mix them and claim those are the best of both worlds, the best of bone marrow-derived exosomes and the best of umbilical cord exosomes. There are two problems. One is scientific. The scientific problem is that it's nonsense. If you have a functional and potent perinatal tissue-derived exosomes, you don't need anything else. Bone marrow-derived exosomes are far inferior, and they are not necessary to actually soup up or improve your exosome product. Secondly, even the term hybrid exosomes is not correct because hybrid exosomes comes from the idea that you fuse a biological exosome and a synthetic one that is filled with, say compounds or drugs, and you fuse them to actually make them better as a vehicle for your drug delivery. So the term is used incorrectly, and it's really unnecessary and not a good idea. And just on the side, it's an American company, and in America, it's actually unlawful to mix two biologics from two different donors. They keep doing that because the FDA has not come after them, but eventually the FDA will notice, and then they will get shut down and will no longer be able to actually sell that product to begin with. Interesting thing. The same company actually recently came up with, they claim to have invented it, synthetic exosomes. They are usually called, as a technical term, this vendor is calling them, and they even claim that they have invented it, which is absolutely incorrect. Niosomes have been around for a long time. And again, they are used as a vehicle for the delivery of some substances, small molecules, compounds, drugs, and they would be interesting for the pharma industry. So if they want to deliver a certain drug or compound, they can create those synthetic exosomes and fill them with this and use it to deliver their compounds. A problem with it is that they have shown to have some side effects because they are synthetic, they are artificial, and the body is not always accepting these kinds of things. But coming down to the point, synthetic exosomes or niosomes are, artificial, and they're empty. They're just empty bubbles, and then you can put in whatever you want. And this one company, they put in 10 select growth factors because they're actually, by default, a growth factor company. So there's nothing wrong with the growth factors. These growth factors are relevant and interesting, and they have some benefits. However, a biological exosomes features hundreds of growth factors, and in addition to that, also lipids and metabolites and very important RNAs. So RNAs are considered the blueprint and instructions for the building blocks, those functional proteins, to actually regenerate and repair a cell. So if you just create an artificial exosome, an iosome, or biosome, and put some growth factors in, that might have some temporary effect, but it's a far cry from biological exosome. And their claim that it is an analog which operates at the same level is just incorrect, wildly incorrect, and misleading customers. Here is just a brief schematic how exosomes actually mediate their signals. They are tiny little bubbles, and then they fuse with a target cell and therefore introduce all their cargo, which, as I mentioned, consists of mRNAs, microRNAs, which are the instructions and blueprints of how to do things, and then functional proteins like growth factors, cytokines, chemokines, lots of lipids and metabolites, and all of which together enable a cell to regain full functionality by being reprogrammed and reset and reboosted. That is true for senescent cells, just like such as hair follicles, for instance. When they stop functioning, you can actually reactivate them. It is true for skin. So if your skin starts, wrinkling, drying out, and these kinds of things, you can utilize exosomes to actually reboot them and reestablish a more youthful state in those tissues, etc. But it does require a functional exosome. Only when you have this intact bioactive bubble that can fuse with a target cell, you have the full benefit of your exosomes. If you have, for instance, lyophilized exosomes where the bubble has burst, it can no longer fuse with the cells. So you might have some free-floating factors around there that can give some temporary benefit, but they cannot reprogram a cell. My analogy for this is imagine you have dry skin and you put on a moisturizer. Then for the time you're using this moisturizer, your tissue and your skin looks fine, But as soon as you leave it away, you go back to your dry skin because the underlying problem has not been fixed. A bioactive and potent exosome can actually fix the problem and establish or make the skin into a healthier self in a more youthful state where it isn't dry. So that's the big difference between a functional and bioactive exosome and those exosomal products you find in the market most of the time. Here is the Harvard study I referred to a little earlier, which gives you an idea, and these are the kind of results that you also want to see from your vendor. So these pictures here are trans electron microscopy pictures where we label exosomes to actually find them. Here is an example of an H0 exosome on the left panel. This is what it looks like. It's a little bit like a donut shape kind of thing. And this is an intact exosome that we actually need to have the full benefit of exosomes and how they work. The other two panels on the left and right, on the right, are examples from the market leaders and their products, and you will not be able to find a single intact exosome at all. And all you find are these aggregates, which are either growth factors or like denatured proteins. All the RNAs get destroyed, so you don't have the instructions, and that's the best you can find. Down the three panels are the corresponding nanoparticle analyses. You see in the left panel that corresponds to H0 exosomes, where you have like one solid peak at about 100 nanometers, which is the average size of exosomes. Panels that correspond to our competitors, you see that there are peaks all over the place. It doesn't really make sense. There is no peak at around 100 or from 30 to 130 nanometers, which is the typical size of exosomes, which just goes to show that there are no intact exosomes in there. And what you find is aggregates of exosomal trash and other things that they might add in the process of lyophilization. Certainly not a good thing, certainly not what you want to have. To summarize that a little bit, these are the red flags that I wanted to point out. Stay away from lyophilized exosomes because there are no exosomes in there. Make sure that exosomes are derived from perinatal tissues and not from adipose tissue or bone marrow. Microvesicles are not exosomes, so if some vendors try to convey it's the same thing, it's not true. Hybrid exosomes are unlawful, not necessary, and not good. Niosomes can have adverse events, can cause adverse events or side effects that you don't want to have, and they are not even close to what a biological exosome is. Conditioned media just means that the vendor didn't go through any trouble of purification or having a defined product. They just throw tissue culture media on whatever cells and try to sell it to you as an exosome product. It might contain a few exosomes, but it's mostly used up media that can even be, a little bit dubious in its application. Particulates, you find that a lot, since when you say you sell somebody 10 billion exosomes, you have to demonstrate that there are really 10 billion exosomes in there and that they are exosomes, which requires the TEM or NTA analyses. So some vendors that don't do these kinds of things just started to sell particulates and don't call them exosomes anymore. They might use exosome as a buzzword to actually get you hooked, but then on the fine print, it's just a particulates, which means that they are not defined, not purified, and you don't actually know what it is, the product. Another red flag is plant-derived or animal-derived exosomes, simply for the fact, as we said, they are responsible for cell-to-cell communication, and only human products can properly talk to human cells, which just makes sense. Just like we cannot talk to animals or plants, they cannot talk back to us. So it's just these two sources are just a convenient alternative to actually derive exosomes or whatever they derive from those sources without getting on the FDA's radar, but they are incompetent and not suitable for the use in humans. Also be aware of single cell line expansions. So there's vendors that advertised that they had one donor pre-COVID and only used their cells for the derivation of exosomes. Again, sounds nice for all those that are afraid of COVID or COVID vaccines. It means that those cells are really old and that they continue to use them over and over again, just like we have to assume our market leaders are doing. They have once upon a time established a cell line and just keep thawing them and reculturing them over and over. The problem is that over the course of culturing, the longer you culture, the higher the risk for contamination is, or for spontaneous mutations and for differentiation. So eventually, these cells are no longer stem cells, and you don't really know what they are or what they. So definitely not a good idea to use those. And the last point, even though it's one of the most critical, if there's no certificate of conformance or electron microscopy or nanosite analyses attached to your product, I would suggest to stay clear of them because then the manufacturer did not go through the efforts to abide by certain regulations and proper manufacturing hygiene and etiquette. That sums it up. If you want to reach out to me personally, you may want to take a picture or copy that barcode that leads you to my LinkedIn page where you can find my contacts, or you can contact me at my AT Venture Center email address. You can also, of course, contact me through our company websites or through HiSam, whatever is most convenient for you. And I'm always happy to answer any kind of question and convey scientific background to help guide you through the process of what is so exciting, but also important about exosomes and the use thereof.
Thank you, Dr. Michael, for your nice introduction. Actually, one of the major questions we intended to do this meeting today is about the safety of using the exosome. So one of the frequent questions doctors asking us that some companies like growth factor companies, they promote that exosomes might cause cancer. Can you please about risk of causing cancer? And then I will open questions to doctors to ask you any questions they need.
Absolutely. So the cancer question, it is mostly derived from, I touched on this a little bit earlier when I talked about the pluripotent stem cells. Embryonic stem cells and iPS cells have been shown to cause teratoma when they are clinically or preclinically applied. So that's of course a concern. Exosomes, however, cannot do that. Even multipotent stem cells do not do that. MSCs have never been found to actually cause or promote cancers. And exosomes will not do that either, much less so even. The risk is even lower. I think the concern or the hypothetical concern is for growth factors. There are so many growth factors involved in exosomes, which is a good thing. And there's a concern that some of those growth factors could also support tumor growth. It is really not the case because it's up to the cell to actually assimilate those exosomes. And they are harvested from stem cells. So by default, they are actually designed to regenerate and repair and fix things and not cause or promote any cancers or tumors. In addition, they do not contain, for instance, DNA. So they have no mutagenic properties or content cargo that would cause anything. So this cancer concern is like not even hypothetical. It's based on something else which is very old and refers to pluripotent stem cells. And I can assure you that exosomes have never been found or observed, not even close, to cause anything like this. So this concern is highly hypothetical and is not substantiated by anything we know about exosomes. So having lots of growth factors is a good thing, and they can only do good if they are derived properly. So it is really important to have a proper cell source and proper quality controls in place.
Great. Thank you, Michael. Any doctors who want to ask, please unmute yourself and you can ask Dr. Michael about any concern. Is there any questions any doctor wanted to ask?
Okay, can I ask a question, please?
Absolutely, yeah.
Yes, please go ahead.
Okay. First, thank you, doctor, for your great information. I want to ask you, how can I know that the real exosome is out of work or it's as expired or have bad issues with using
Um,
during storing?
mhm. So we have a full quality management system in place. It's unfortunate that Erin, our CEO, could not join us today. She is by training a quality engineer, and we don't like have just a little lab tucked away. Mentioned that we are deeply rooted in regenerative medicine. So our parent company, Regenerel, which is manufacturing exosomes for medical applications, is actually the prime manufacturer, and everything is manufactured according to clinical grade standards. So we abide by all the FDA regulations and laws, and then some. We have New York Tissue Bank license, which is in the United States one of the hardest licenses to actually get. We have extensive quality controls and third-party testing
I
that
know,
are.
Doctor, you are the best. I'm not speaking
Mhm,
about your company. I'm speaking
mhm.
about during the saving in the clinic.
Okay. For as long as you store and handle those exosomes properly, there's really no concern. So they're extremely stable. They have been shown to have excellent integrity. There's a little bit of a confusion that Dr. Haizam pointed me towards in terms of expirations there, the dates and stability. So we always have like a recommendation how long you should store them and how you should handle them. And then there's an official expiration date, which is far beyond what we propose or suggest to use them in. So for as long as you store them properly, those exosomes are safe and stable and very functional. We have shown even storing them in a refrigerator rather than frozen, they are stable for actually more than a year. It's nothing
Great.
that we recommend because there's a difference between storage and handling, but they're supremely stable and therefore safe to use for a long time.
That's good. Very good.
Doctor, there is another question. One of the doctors sent us on the chat that she said that we use H0 exosome post
Mhm.
lasers topically. Is there any
Mhm.
chance a patient can be allergic?
Hardly, because they also don't contain allergens and they are so small. If, for instance, you are talking about stem cells, there's always the risk that a patient who is very sensitive could react to stem cell therapy because stem cells are actually living entities and they are much bigger. So the body will recognize them as something foreign. However, exosomes are so small and they do not contain any allergens, which is why we are using them. So they don't have surface markers that would uh, have the body identify them as something foreign. And they are so small that you can apply them even in large numbers without causing any concern. So if you happen to observe any sensitive reactions around this, it's most likely that it's from the adjunct treatment like laser or microneedling or so, but it's not coming from the exosomes. They can actually help to fix whatever problem arises, like inflammations will be addressed properly, you heal faster. We have not seen any adverse events or side effects in the use of exosomes around these treatment options.
All right. Another question from Dr. Matthew George. How many sessions we need from exosome is needed to produce an appreciable results?
Mhm. That is a moving target, I have to say. So in the field, there are no clearly established benchmarks, and then it depends on what you address. And then, of course, it also depends on the patient, what indication you try to address, how severe it is, how old. There is like something when we talk about rejuvenation, for instance, skin rejuvenation, we have found that younger patients, so in their 30s, respond better to the exosomes or more quickly than elderly patients in their 60s or so, which of course is a little bit disappointing for the elderly patient, but it also makes sense. So if you talk about rejuvenation, let's assume you apply those exosomes and you shave off 2 or 3 years. If you do 3 sessions on a 30-year-old and they look like a 20-year-old again, that's a noticeable difference. But if you're in your 60s and you shave off 9 years, you might not see such a big or drastic effect. So it takes a little bit longer. So the answer to your question is, unfortunately, it's very individual, and it's a little bit by trial and error. We have also found that more is better in terms of higher numbers of exosomes might give a better result. But it's also put into relation, because if we use 5 billion exosomes on your face after microneedling versus 50 billion, you don't get 10 times the benefit. So you might want to do higher frequency, that you do that every 4 weeks for like 3 times, and then maybe 2, 3 times a year. That would be a reasonable frequency. If you have like more concrete indications or applications to me, and I would address that in more detail, what I would suggest or what I know about these particular applications. So it can be
Dr.
very, very
Most of
different.
the attendees today, aesthetic dermatologists. So the indications like acne scars, post-surgical scars, rejuvenation, pigmentation, hair
Mhm.
loss. This is the indications doctor will be interested to hear the protocol about it.
Mhm. Yeah, I usually, whenever it comes to skin, doesn't really matter what kind of indication it is in particular. There's always the suggestion to do like 3 treatments in a short period of time. Let's say like with a month in between 4 to 6 weeks, 3 treatments, and then see how well they respond. And then you can adjust the numbers either up or down and the frequency. So we see very good benefits after 3 treatments, and then you probably do like once or twice or 3 times a year for maintenance. But age and the severity of any given condition is, of course, a factor as well.
All right. So 3 sessions,
Hi,
one.
I have a question. I'm Dr.
Mhm.
Shazadi. Regarding thank you for the lecture. So my question is, as you said that, is there a limit for the amount of sessions? Like, is there, because some products you cannot use after a particular number of sessions. So is there a limit to it? Can it be, can it be up to 10 sessions or 20 sessions, or is there anything that we have to worry about in that sense in terms of toxicity or something
Mhm.
like that? That's one. I have another question after that. So if you can just answer this one, then I'll ask me my other question as well.
So there's certainly nothing to worry about in terms of toxicity or doing too much. Doing too much is only that it becomes a little bit redundant, maybe, if you do it too much, too often, too fast, that you don't get the full benefit. An example, for instance, hair, like the turnover for hair is like, I think, 3 months or so. So if you have a hair treatment, it wouldn't make sense to do it every week. It would probably be a waste. But this is like the only concern, that you actually waste benefit. You don't run into toxicity problems. And it's maybe also a money issue, like how much you want to pay for the treatments. But long story, short answer, no toxicity worry. And the intervals and frequency and the number of exosomes to apply in any given treatment should be reasonable, you know. Does that make sense?
Yes, that makes sense. Thank you. It was actually regarding a treatment for hair. That was what was my concern, because, as you said, it takes about 6 months for us to sort of like achieve, and especially like depending on the severity of the case, you might have to do more sessions than what is normally recommended by, when you say three sessions. So what I normally do is I combine it with PRP, which I'm doing at intervals of, let's say, 3 weeks, and then exosomes is at 4 weeks. So that's how, I'm doing it. But I don't know if that's the right way, or is there anything that you would like to add to that?
I always say if you do exosomes, you don't really need PRP, because exosomes are 100, if not fold more potent. But I'm
Correct.
acutely aware that some patients started with PRP and they like it. There's some benefit, there's some progress, and then they want to combine it to actually boost the effect. So there's no reason why you shouldn't do that.
I
You can.
think yeah, yes, I understand. The only reason why I prefer, I mean, why I'm doing it is because, as it is not recommended for us to inject them. So I just feel that because PRP is something that we can inject and safely inject it, in exosomes, I can only apply it after microneedling. So, that's what my, so I do a combination just so that I cover both bases mostly. But of
Mhm.
course, I know that the exosomes are probably doing much more than the PRP, but it's just, in my head, I'm
Yeah.
just thinking that way. I don't know if it's the right way, but that's just what my sort of thought process in the whole thing is. So,
Absolutely. I think it's a wonderful approach because you really tap into both options. Uh, Resilier exosomes can officially not be injected. That's correct. Issue because it's labeled as a topical cosmetic and therefore can only be applied topically. Injection, any kind of injection is considered a medical application.
yeah,
And you can do that too, but that would be under the Regenerel brand. We know of very successful cases in Pakistan where the regulatory is not an issue. So they can use our topical exosomes and inject them. And we heard of many cases where they actually mix the PRP with the exosomes and inject them and have wonderful results. So that is an option. It is officially not allowed with the Resilier exosomes.
yeah.
They would be suitable for injection, but that would be a Regeneral brand. So we have to strictly separate it as a manufacturer, cosmetic applications and medical applications.
Okay, thank you. Just coming to the next question quickly. I'm sorry I'm taking up your time, but this is regarding the storage as well. So it's always a concern that, for example, if I'm treating a small area and I've used about 2.5 ml of the solution and I want to store the other 2.5 ml, what I've been told is that I can keep it in the fridge for about 3 months after, but somehow I just don't feel very comfortable doing that. So I just would like to understand, because as I understood, what you said is that we can store them for up to a year after we take them out of the -20, correct?
Correct, yeah.
So, which means that, because it's a little bit of a conflict in my head. So, we
Mhm.
have these exosomes which are at -20 in the clinic. Once
Mhm.
we defrost them, whether I use it or not, I can keep it in the fridge for 1 year. But
Mhm.
if they're at -20, they expire in 6 months, even though the
Mhm.
date on it is, let's say, 2026, but they
Yeah.
expire within. So I would just like to understand why there is this sort of a discrepancy, and it's kind of like I'm not being able to put
Mhm.
my head around it. So if you could just explain
Absolutely.
that, please. Yeah.
So I'm very sorry that we caused that kind of confusion. There is something that we state on the records, and then there's information we can give you off the records. So as you said, like if you thaw the vial and you use some of it and put it in the fridge, you could store it in there for like 3 months or a year without loss of stability or activity of those exosomes. But you said that you wouldn't feel comfortable really, and I share that sentiment. I wouldn't feel comfortable about it either because I always, in my mind, want to um, abide by best practices and use them as fresh as possible. So
Yes.
long-term storage is never a good idea. This particular example, if you feel like you only use half of it and you don't know when you want to use the other one, you can even refreeze them. We just do not recommend repeated thaw freeze cycles. So if you feel like you cannot use it within the next 3 months or something, freeze it away again and then thaw it the next time comes around. So there is a best practice recommendation. That's why we say like in the fridge, 3 months, in the freezer, 6 months. They do have an expiration date in June 2026, the current lot that is sold through
Yes.
Lifetime. That is the official expiration date that we put on from the date of first manufacturing and all the extensive testing that we are basically forced to put on. That doesn't mean that You couldn't use them beyond that, but I also don't want to complicate things. This is like by FDA, MOCRA guidelines, you have to put an expiration date on it. And as a manufacturer, we would say like beyond that date, you should not use them anymore. That the other things are so maybe vague or confusing is just a matter of what we recommend as best practices versus what we have found, because we do ongoing testing and we know that our exosomes, even if you store them just in the fridge for over a year, they are completely stable and efficacious. But we wouldn't want to recommend that as a practice, Therefore, keep them frozen until you're ready to use them. If you don't use them up and they're already thawed, keep them in the fridge. If you're concerned that you won't be able to use them anytime soon, refreeze to go around it. And it's just to give you the comfort and the assurance that even if you push it a little bit to a longer term storage, there's not a problem with the exosomes per se.
Okay, so which means that if I receive the exosomes and they are frozen at -20 for 6 months, they're good to go. Otherwise, we need to sort of, um, I need to get a new batch, or is that's what we're talking about, right?
Yes, exactly.
Okay.
So, I mean, chances are if you order a batch, some of them will be used. So if you have like one or two left over at 6 months, they should still be fine.
Okay.
The recommendation is just use them as quickly as possible.
Okay.
Because there's also not really need for long-term storage, rather order in smaller batches and more frequently, because then you always get the fresh exosomes.
Yes. Okay. Super. Thank you so much. Thank you.
Absolutely.
Dr. Hickey, we have a few questions in the chat. Compare with plant exosome or milk exosome. How do you compare Receliel H0 to milk or plant exosome?
So people are very inventive coming up with alternative sources of exosomes. So as I mentioned, you find exosomes are secreted from virtually all cells. Milk is something new. That's actually a funny one. But if you want to look for the most potent exosomes, it has to be perinatal, human perinatal tissue. As I said, placenta, umbilical cord blood, umbilical cord, Wharton's jelly, maybe amniotic fluid. Among them, Wharton's jelly is the most potent and safest source. Everything else, other than perinatal tissues or Wharton's jelly in particular, is suboptimal. If they are derived from salmon DNA and any plant stem cells and so on, it's really all rubbish. It's just people that want to take it easy, or they try to market you into this being something fancy or good. I am aware that people started deriving the market leader, actually. They also derive exosomes from roses or orchids. It's complete nonsense. It's just like try to tickle somebody's fancy, but it's not an alternative to a very highly potent perinatal tissue-derived exosome. So that's the long and the short of it. Go for Wharton's jelly, no matter who the vendor is. Anything other than Wharton's jelly is suboptimal and sometimes even concerning. And anything like milk or plant or animal derived, they cannot talk to our cells. So they are like useless. They might have some superficial benefit, but they're really not exosome suitable or able to reprogram cells and really regenerate and rejuvenate.
Yeah. And what about temperature and storage? Can you summarize again what temperature you are storing in the lab, and we are here storing in -20, because the question again about this.
About the storage, I didn't get completely this.
Temperature and storage.
Yeah, the storage. Again, we give out recommendations to store them and for not too long, because we want to encourage to use them. But storage is actually not a problem. -20 is fine for a long time. Even the fridge is fine. And we were prompted by Dr. Haysam that this might be a little bit confusing. So we're trying to summarize a white paper that explains it a little better, why there are different dates and recommendations. And as soon as we have finished that white paper, I'll be happy to provide it, that everybody can look into it. And then keep prompting me if.
All right. Another question from Dr. Andrew. What are your thoughts on injecting exosomes for orthobiologic use? Example, knee OA.
Mhm. It's actively being done. It's great. It works. It's very effective. It still requires a little bit of dose finding, and sometimes the best route of application. We have also seen exosomes being applied systemically as like an infusion, and in addition to injections directly into joints, etc. The only thing that I have to mention is it's a different brand. That would be Regeneral. The exosomes are the same quality for both applications, but whenever it comes to medical applications and injections, it's just a different brand. It's Regeneral, same exosomes, all the same standards, clinical grade, and we have used them plenty here in the US under a right to try, and they have been shown to be extremely efficacious. And it's a wonderful new step of stem cell therapy, really. And it's so much easier. And depending on what the indication is, we've seen them applied in neurological problems. They're easy to cross the blood-brain barrier, and so therefore very effective. Many medical applications are possible. And orthopedics, we know for a long time that they're, very, very prevalent in using either stem cells or exosomes. So yes, can be done, highly recommended. And it's just a different brand. So because it's a medical application.
All right, thank you. Another question from Dr. Fadi Al-Sabbagh. Is there any protocol how to use exosomes for treating hair loss? Also, can exosome mixed with PRP in the same session?
We have touched on that before. There's different adjunct treatments that you can combine with exosomes to make it more efficacious. Microneedling is one of them. You do a microneedling on your scalp and apply the exosomes thereafter. A wonderful system is, something like the CO2 guns, where you can apply them directly, very, very effective and less painful. DEP is another one. Certain laser treatments are amenable to be used together with exosomes. So all of these things work. The benefit or the preference of those are these are treatments that are then followed by exosome applications. So that would be considered topical anesthetics. PRP, combination of PRP and exosomes, as I mentioned, I think it was Dr. Shehzadi who asked that question. Very effective. Thing is that I think in the UAE, it's not possible to inject the Riselier exosomes. So you would have to resort to Regeneral sourced exosomes. But yes, the combination of PRP and exosomes are really effective and highly recommended. It's just considered medical application again, so has to be sourced from Regeneral.
All right. One more question from Dr. Florencia. The most serious and clear explanation of exosomes age 0 has FDA approval? She's asking, do you have FDA approval?
That's a tricky question. As you might know, that's probably why that question is always being asked. The FDA is hesitant to approve any kind of stem cell or exosome treatments. There's various reasons for it. I think it's very hard to govern it. They have a hard time to come up with a framework of what stem cells or exosomes should look like, how they should be manufactured, and so on, to even consider them for approval. And secondly, there is a big pharma lobby behind it that undermines every effort because the pharma industry is not very happy with either stem cell or exosomes because these two agents have the potency to cure diseases, while the pharma industry wants to treat diseases and not there's more money to it. So that's the background. But I can give you a little bit of a story. As I mentioned, we are deeply rooted in regenerative medicine. So what we did first was we set out to create an off-the-shelf solution for stem cell therapy. So we manufactured those MSCs, and everything we did in cooperation with the FDA. So we contacted them proactively and said, this is what we want to do. And we don't want to be like on your radar in a bad way. So we wanted to be very transparent about it. And they were surprised that somebody actually made an effort to contact them before they started doing stuff. So they gave us all kinds of advice, and we abided by all the FDA regulations and quality laws and so on. In this, we established those MSCs as a off-the-shelf solution for stem cell therapy. And then we sent an application to the FDA to use those stem cells to treat a patient suffering from traumatic brain injury. It was a very bad case. The patient had no recourse of treatment, was in a vegetative state, and would have been a hospice care kind of case. We applied what is called expanded access. It's certain mechanisms where you can apply for one treatment, for one patient, for one indication. And we made a case what we want to do, what we would expect as an outcome. And given that we had a good report with the FDA and they knew of our quality and quality measures and safety and everything, they gave us an approval to do that within 24 hours of our application. We treated the patient, and the patient fully recovered from traumatic brain injury. So what we have is an FDA IND approval under expanded access for the use of our stem cells to treat patients with traumatic brain injury, which is a first in the US. So in this sense, we do have an FDA IND approval. It's just very limited. You can only use it for a particular indication, etc. But it gives you a sense of the quality and the safety of our stem cells, and we use exactly those stem cells to generate our exosomes. So by association, you kind of have an FDA stamp of approval of sorts. It's just not official, and they will never do that for a long time to come, I fear. But things are in the works. So we're also working on lots of clinical trials to apply our exosomes for various conditions, and we will get there. It will just be a long time. These things typically take 5, 10, sometimes 15 years, and the FDA is not very much in a rush to approve anything. So we are on it, and we are very close, but it will still take time.
All right. Any contraindications? How long do the result of exosome last?
Contraindications, none. I would always be hesitant. We say like no pregnant women. Those are the standards, right? Standard exclusion. But just because of a hypothetical concern. No patients with active cancer for the cancer to address the cancer concern, but it's not really contraindicated. We just don't want to do that, like pregnancy, active cancer treatments. These are the things. Steroid use might render exosomes ineffective, and therefore, in this regard, they are contraindicated. It might be a waste. But other than that, there's no contraindication really that I can think of. How long does it last? It really depends on the indication, the kind of treatment, and so on. You have to imagine exosomes can actually address age-related issue like hair loss, for instance, or our skin becoming less supple and so on. So they can address this and fix this, but the underlying process of aging continues. So therefore, once you got this fixed, you still have to maintain it which is why we recommend, 3 skin treatments in short order, and then 1, 2, 3 times a year to actually maintain this kind of stuff. So aging still progresses, as diseases often progress still, and then you actually have to address and do some maintenance. I hope that answers that question.
Yeah, clear. Thank you. One more question. Very nice question, I can say, about from Dr. Iman Uqba. Is there different exosomes for cosmetic treatment, like one for hair, one for rejuvenation, one for hyperpigmentation?
Very good question. Yeah, I like that question too. Yes and no. So the exosomes are all the same. What differs and what we actually might introduce shortly into the market is different concentrations and numbers for different applications. To give you an idea, if you do a face microneedling and exosomes after, we found 5 billion are sufficient, and the ideal volume has been found to be like about 2.5 milliliters. That's why we have those sizes. If you do a larger area, you want more exosomes, and then the volume to number of exosomes might change, or if you do scalp treatments and so on. So we will change the volume and or offer particular volume and concentrations for particular applications. So in this regard, yes, there are differences, or there will be, but it's always the same exosome. They just come in different numbers, or will come in different numbers and different concentration or different volumes.
So there is different concentration, but there is no specific exosome for hair, specific exosome for rejuvenation or scars, like because
Yeah,
it's
that is not necessary. Yeah, because we just need these stem cell derived exosomes that have the full potential for regeneration and repair. And then the tissue might be different, or the hair, hair follicles, but they can utilize those stem cell derived exosomes for their own purposes. So they don't need to be designed for particular indications. They're like omnipotent, so to say.
All right. Clear. From Farah, Dr. Farah, there is a question asking about is there any study show carcinogenic effect because it's from human to human?
Mhm. It's always hard to prove that there is nothing. You can prove that they cause something if they do. There's a nice paper that I can send to you, and you can maybe distribute it, that actually investigated this, and there's no carcinogenic effect observed by exosomes. They're considered safe.
All right. Clear. Any more questions from doctors to address to Dr. Hiki before we end the meeting?
Yes, please, Dr. Haitham. I
Yes.
got some question from the doctor related to injection oxosome with Botox and filler. Can we mix it, the injection with Botox or filler?
I would argue you can replace Botox and fillers with exosomes. I find them to be as effective if used properly, and they are better because they actually fix the problem rather than just a palliative remedy. Whenever it comes to, I wouldn't argue against it. So if that's something you wanted to do, inject them together because people love Botox and love fillers, and you want to add exosomes, there's no contraindication really. Theoretically, could. Although I have to say it's injection again, therefore, Resilier brand, we must not do that. If you want to have exosomes that you can inject, that would be a Regeneron brand. But on a scientific or application level, there's no contraindication, so you could if you wanted to.
So Dr. Hiki, regarding the injection, most of the doctors, they prefer to inject to guarantee the delivery, more than the microneedling.
Mhm.
We tell the doctors clearly that the registration of Resiliel is topical microneedling, but is there any
Mhm.
problem if they inject the Resiliel like intradermal?
There wouldn't be a problem as far as quality or safety is concerned, or potency even. So, as I mentioned, they are clinical grade. So, by design and by manufacturing standards, they can be injected. They would be completely safe, and we know them to be efficacious. As a manufacturer uh, and representative of Resilience, that you must not do that. I cannot control it. So scientifically and medically speaking, you could inject those exosomes. Regulatory wise, it is not allowed.
Right. Dr. Paula, she's asking, it can fix wrinkles, like can exosomes fix wrinkles that need Botox? I'm not sure about that. We can't.
Mhm. It can over time it will, because it has the ability, even though I don't like the word rejuvenation because it's so overused. Exosomes have the ability to do just that, to actually reprogram cells into a more youthful state, and therefore your skin gets younger. It requires some initial applications and then some maintenance, but we have seen that we have done. I actually can go through. There we go. I hope you can still see that slide.
Yeah, we can.
Um, we did a 100-person skin rejuvenation trial, and those are the results that we also published. That's the feedback. Pigmentation was improved 89%, firmness 88%, wrinkles were improved 86%, redness, it was after microneedling 85% approval, texture 90%, and radiance was 92.6% improvement. These are, of course, subjective measures. It was reported back after 3 treatments of microneedling plus exosomes. I can assure you, though, they were blinded, so people responded and gave their subjective impression whether they had been not improved at all or somewhat or strongly. So we vetted them. So they were not, as I said, it was blinded. So they could say anything. People didn't have to tell us just to mollify us that the results were good. So these are real life, real application results, and they were quite impressive. So if you talk about wrinkles, 86% improvement. So I would say that is a yes in the vast majority of cases. And sometimes it's just about maintenance of those treatments to see even better effects.
Yeah. So again, there is a question why Regeneral brand is different.
Mhm. The product is not different. Here in the US, there's a law you cannot provide products for both medical applications and for cosmetics under the same company umbrella. So that's not allowed. That's why we created the Resilier brand, which is just topical applications in the field of aesthetics and cosmetics. And then we have the Regenerel brand, which is manufacturing the exosomes and also stem cells for any kind of medical applications. So Regenerel is the parent company. So they produce all the exosomes. So even the exosomes that we sell under the Resilier brand are manufactured by Regenerel and therefore abide all the FDA regulations and are clinical grade. So the exosomes are in fact identical, but one brand you can use for aesthetics and cosmetics, and the other one you can use for medical applications. It's just a regulatory necessity to strictly separate the two. That's why I said our exosomes, Resilier exosomes, you could inject them quality and manufacturing wise and so on. They're identical with the medical application exosomes, but under the Resilier brand, you're not allowed to do that. So I hope that answers that question. It's just a regulatory requirement to separate.
Yeah, clear. Thank you. Again, one more question from Dr. Mariam about the depth of microneedling should be used with exosomes.
Mhm.
Should it be deep so that we have some blood, or it can be superficial, especially if there is pigmentation? They should go deep with the
Yes,
needling or only superficial?
it can be superficial. The penetration depth is not really a strong issue. The exosome have been shown to even like penetrate the skin without any adjunct. Um, the beauty of microneedling is that you do that the exosomes can penetrate even a little bit deeper, and you also cause micro damage and micro inflammation by the application, which is a trigger for the exosomes to become activated and also for the cells to start regeneration, right? And then they take up all those exosomes that are supplied, which boosts the effect and the regeneration process. The depth is not critical, and I wouldn't go too deep anyway. So, for instance, if you are too radical with your microneedling depth, you might, for instance, damage hair follicles, and then you actually render the benefit that exosomes could offer obsolete because you damage the hair follicles to a degree that they cannot really just get rebooted by the exosomes you apply. So, short answer is the depth is not critical, and superficial will do. You don't have to achieve pinpoint bleeding to actually get a benefit.
All right. One more question from Dr. Paula. Can you compare with ASCE exosomes?
Yes, I can. ASCE has no exosomes. They lyophilize their product, therefore destroy the exosomes, and you just have exosomal debris. So, as I pointed out during this lecture, they cannot function properly. So, you need an intact bioactive exosomes to fuse with a target cell and therefore enable the cell to actually reboot and regenerate um, and be fully functional again. ASCE is not able to do that. They lyophilize and destroy the exosomes, and what you have left is aggregates of some proteins. So all the RNAs get destroyed, so the cells would not get the instructions of how to reboot and regenerate. So all you have left is really some residual growth factors that might have a temporary and very superficial effect, but it's very short-lived. And to be honest, it's a terrible product, and they kind of know it.
All right. Can exosomes help with aging, eyesight decline? Eyesight decline.
Something, yeah, that's something that's currently under investigation. We actually do entertain a study. It's not clear yet. There's a promise that it will, and we do a very simple trial where we use exosomes as eye drops and see what we can achieve. This is a black box right now, so I cannot promise anything, but there is some initial indications and publications that indicate it could work. It could work fine. The best route of application has to be determined, and we have to see to what degree we can actually remedy these indications. But it's a very interesting topic, and it's currently pursued as a clinical trial.
Yeah. If you have no preservatives in the vial, how does it last so long?
Well, you don't open it. It is filled aseptically in a clean lab under CGMP. So everything is completely sterile, and we do sterile testing. And as I said, we also have third-party testing. And these vials are, pharmaceutical grade vials when they are filled. So what you have is only saline and pure exosomes, and that's what they remain. So unless you take the cap off and expose them to the environment without a lab bench, they will be sterile and aseptic and clean. So there's nothing to worry about. And they don't need stabilizers either. So that's it's a very interesting fact that we have established.
All right, great. Back to what you mentioned earlier, PRP and exosome is very powerful treatment. I was treated 5 years ago with this protocol, injections and IV, also with the exosomes, and it completely healed all of my nerve damage in my lower spine area. It was the only thing that healed everything finally. I have had zero pain ever since.
Wow, that's wonderful. That's a nice confirmation and testimony to hear. I believe that exosomes are extremely powerful, and we have only begun to actually tap into their full potential. And it will be very exciting to see what we can achieve, not only in the field of aesthetics and cosmetics, which is very close to all of us, of course, as we age, but also medically. It's extremely promising and very exciting.
Yeah. Dr. Hiki, this is the last question, and we will end the meeting after. Why you think it's difficult to make synthetic exosome? There is vendor who claim that they make synthetic exosome and they copy what is inside exosome and they make it artificially in the lab. Why you think something like this is not possible?
It's not at all possible. If somebody claims that, it's just flat out a lie. Synthetic exosomes are extremely easy to produce, but they are synthetic and they are empty bubbles. So everything you want to put inside, you have to actively put inside. So you have a synthetic exosome, let's call it exosome, it's niosome. Then you have synthetic growth factors that you derive from somewhere to put in there. And one competitor, which is the second market leader, they actually put, I think, 10 growth factors in there, and the growth factors are good, relevant, but a biological exosome has hundreds of growth factors and like RNAs and
Dr. Hiki, are you still there?
give is like an artificial flower. If you have an artificial flower and you look at it from afar, it might look as real, right? But when you get closer, it doesn't look anything like a real flower. It doesn't have the beauty, it doesn't have the smell, it cannot pollinate, it cannot grow, it cannot wilt. There's like nothing biological to it. And somebody who claims that any synthetic exosome or niosome or biosome is similar to a biological exosome, it's just inaccurate, untruthful, misleading, and I would argue it's customer fraud if they really claim that. So be aware, synthetic exosomes are not the real deal, and they are nothing like biological exosomes.
All right. Thank you so much, Dr. Hiki, and thanks everyone for attending the lecture. And anytime any doctor want to reach to Dr. Hiki, we can provide you the direct contact to email him for any questions or concern you have with exosomes.
Absolutely.
Thank you, everyone. Thank you so much.
Thank you for attending. It's my pleasure.